This passage is about tea DNA in tea leaves. Very interesting article. Normally, science and technology articles are easier to read, but there's a lot of information given in the passage. So, for the questions, if the questions are direct questions based on information given in the passage, a good idea to go back to the passage, refer to the relevant portion of the passage, and answer the questions. If you rely on your imperfect memory of what you read, always remember you read the passage once quickly but thoroughly. Try to understand the organization of ideas, what happened, what the author mentioned first, what did he mention next, what did he mention next. Once you do that, you don't try to remember every fact, every sentence, every word written in the passage. So if there is a question on what did the author mean by this, how many species, what DNA did to that, or what plant was used for the experiment, there were 2-3 plants named. So some other plant was named in some other connection. Then you should not rely on your imperfect memory and say, yeah, I think he meant mint leaves or whatever and mark it. You have to go back to the passage, go back to the relevant part of the passage, look at the numbers, make sure the numbers are right. Look at the names and make sure the answer option you're marking is the right one, right? Okay, let's quickly look at the passage. So he basically says that a team of scientists has discovered a new way to uncover some of the interactions between plants and animals. How did they do that? They bought teas and herbs from a grocery store and tested the dried and packaged leaves for minute remnants of DNA, a method called environmental DNA or eDNA analysis. So a group of scientists wanted to see the interaction between plants and animals, and they found a novel way to find out this interaction. What did they do? They collected teas and herbs, packaged teas and herbs from stores, they got it, and then they did what is called an eDNA analysis or environmental DNA. They checked it for environmental DNA. What did the team find? The team found traces of more than 1200 different arthropod species. Arthropods are insects or tiny insects, organisms without a backbone, right? Invertebrates, they're called. So you don't have to know that. They found 1200 different arthropod species. Know that arthropod has to do with animals, insects, from their analysis of just 4 plants: chamomile, mint, tea, and parsley. So they analyzed 4 plants. How? By taking packaged products, and they analyzed DNA, environmental DNA, and they found 1200 different arthropod species, DNA of 1200 different arthropod species in just 4 plants. All right, that sets the tone for the passage. The team had to develop methods to extract and amplify the arthropod DNA from all plant material. The vast majority of the DNA in tea leaves is from the tea plant itself, obviously. Probably 99.999% or something like that is plant DNA. Only a tiny fraction which is left is insect DNA. So 99.999% of DNA is tea or whatever plant DNA. The small 0.001% that is left is the animal DNA, which of course is good, because when you drink tea, you don't want to think that you're drinking insects. He adds that even a minute presence of arthropod DNA is a good sign that the tea isn't dripping with pesticides. My brother works in the same field, and he says, I go to buy brinjals from the market, aubergines, eggplant, whatever you call it. He says, good if you have insects in them. And I would look at him. He says, that shows that there's been no insecticide or pesticide used there. So it's the same way here. He says, when you find arthropod DNA in tea leaves, in mint leaves or whatever, it's a good sign. That means that there isn't too much pesticide that has been used on it. The researchers figured out how to isolate arthropod DNA by finding a key sequence. So how did they do this? Is given the method that they found a key sequence that differs between arthropods and plants. The team discovered on average more than 200 different arthropods from each tea sample. On an average, there was more than 200 different types of arthropods in each tea sample that they found. Not all of these animals could be matched to known species. Some of the animal DNA, they could not match the DNA with any of the known species of animals, highlighting a need for further research on the more obscure and understudied groups. But those that were identified generally matched the known distribution of both the plants and the arthropods. Many of the species, they could not find the arthropod. What arthropod does the DNA belong to? But the one where they found aligned with the geographical location and presence or distribution of both the plants and the arthropods. For example, now this example he is giving, what does he mean by that? Mint tea contained DNA from insects found in the peppermint growing in the Pacific Northwest region of the United States. So they found mint tea had DNA from insects which are found in the peppermint growing Pacific Northwest region of the USA. That particular insect is there, and they found mint tea had a DNA of those insects. Whereas green tea contained DNA from insects native to East Asia. All right, next. Being able to analyze eDNA from commercial teas could make it easier to collect data on insects from around the world. So when you find tests, you find which insect's DNA is there. It will also tell you more about insects around the world. Oh, this is found here. So this insect must be thriving in that particular region, right? According to Eva, a molecular ecologist who was not involved in this study, a common problem in many DNA studies is the limited volume of samples that can be obtained by a small team of researchers. Now she's told the findings of the research, told the advantages of the result. Now to come to the problems. She says one of the problems is you have a small number of researchers. So how do they find so many samples? So the samples they can collect will be only a limited volume, right? Because how many samples can they collect? A small team of researchers. Using commercially produced teas and herbs circumvents this issue. It nicely solves this problem by going around it. It finds a solution to the problem by taking advantage of the existing infrastructure for harvesting, drying and transporting plant material. Tea is collected for commercial use. Tea is collected from different sources, harvested, done everything. So what does the researcher has to do? His problem of collecting samples is taken care of by just buying the commercially produced tea, the packaged version. You could even say that sampling has been done to some degree unwittingly by these companies that have produced those products. So companies will source products from different parts of the world. And so they've got a very nice representative sample. They'll get the best from every different part of the world. So by buying that tea, the researchers' problem of collecting samples from different parts of the world has been solved. So the problem of sampling, because sampling has already been done by the commercial producers of tea or mint or whatever. Analyzing the arthropod DNA from the tea leaves or other dried plant material could help scientists track the spread of insects that are considered pests. Now, another advantage, so this is the problem, they have the problem of sampling, and he has told you how they have overcome the problem of sampling. Second one is, what is the other advantage? One advantage, they said, you will know which insects grow where and their distribution. The second thing is, another advantage is it will keep track by knowing, okay, this insect DNA has been found in this tea which grows in this area. They also will track the spread of insects that are considered pests. Which insects are considered pests? Ones which cause harm, right? A pest is a harmful insect. Insects are often inadvertently, involuntarily, by mistake, accidentally transported around the world, catching a ride in cargo ships, potted plants or firewood. So when you carry a potted plant, by mistake you carry an insect also from one place to another. Or cargo ships, right? There may be an insect on a ship, and when the ship goes from one place to another, the insect is also traveling along with it. Some species relocates, while most won't survive the trip, some species relocate successfully and go on to wreak havoc on forests or crops. Many of these insects which are transported accidentally from one place to another don't survive. But some do, and when they do, they go to that new place and they cause destruction and devastation and those wreak havoc. They cause a lot of problems to the plants and forests and crops in that area. So, what will being able to detect these species do? Being able to detect harmful species soon after they show up in a new area could help kickstart the management process. So, you can check the pest, you can do pest control, you can check the pest population by knowing, oh, this particular DNA has been found here. That means they've just managed to reach that place. So, let's control, let's manage so that the problem doesn't go out of hand. Question 9. Which sequence of words best captures the narratives of the passage? A very common question in CAT. Which of these words show the flow of ideas as expressed in the passage? So just remember summarize the main points of each of the paragraphs. Started by first talking about interaction between plants and animals by doing a study of eDNA. How did they do it? They bought packaged tea and leaves, and they did it, right? Second paragraph, what did he say? He said, and when they did it, what did they find? They found 99.999% is tea, but the rest was arthropod DNA, right? Then he talks about the advantages or disadvantages. Sometimes it becomes difficult to identify. So you need more research to find out which species of arthropod does this DNA belong to. And then he talks about the advantages, right? By identifying, so along with arthropods, there are some insects, some arthropods which are pests. So if they find out through eDNA that this particular species has been found there, maybe if they check the presence of a new species in a particular area quickly enough, then they can manage the pest without the pest problem going out of hand, because these pests can cause a lot of damage to crops and forests, right? So the advantage of eDNA analysis and how it's going to help, right? That was the flow of the passage. Now let's look at the options. eDNA analysis, okay, arthropod DNA, arthropod identification, sampling, pests. eDNA analysis, arthropod DNA, arthropod identification, sampling, pests. What is missing here? Didn't talk about how this happens. Interaction between plants and animals, tea leaves, none of that. Only arthropod part of it has been given. How did they come about it? So it's missing a major aspect of eDNA analysis. eDNA analysis of tea and herbs, that has not been mentioned. So A cannot be. Teas and herbs, all right. eDNA analysis, all right. Extraction, research, pests. What is missing here? Could be tea and herbs, yes. eDNA analysis. Problem is the exact opposite of what is in A. Arthropod DNA has not been mentioned. Yes, they took tea and herbs and did DNA analysis. What did they find? They found arthropod DNA. That has not been given here. What was the purpose of the research altogether has not been given here. So this misses out the arthropod DNA part of it. Cannot be my answer. T, arthropod DNA analysis, future research, pests management. Vague, slightly vague. Which is better? Tea leaves, arthropod DNA, they analyze the tea leaves. Did eDNA found arthropod DNA, identifying it helped them, the sampling problems that they face, how it is taken care of by the commercial packing, and also talked about how pest management can be done. So which is the best option? Our best option is D. Clear? Remember the main points of each of the paragraphs.